Welcome to this new episode of the Virulent Vortex, a podcast in which we discuss infectious diseases across scales, from pathogens and molecules to populations, pandemics, and everything in between. My name is Jaap de Rode. I'm a professor of biology here at Emory University and also the host of today's show. And with me today is Mirko Paiardini, professor of pathology here at the Emory Primate Center. Mirko, nice to have you on the show.
Thank you, and thanks for having me.
Yeah. So a lot of your work until recently has focused on HIV and AIDS. And I wondered if you can tell us a little bit about that work and then how that work transitioned you to think about SARS-CoV-2, which we have started experiencing during the COVID-19 pandemic.
Yeah, sure. So I'm at the Department of Pathology, School of Medicine, but I am based at the Emory Primate Center. Mm-hmm. So my research has been heavily focused on HIV infection, HIV pathogenesis, and also HIV cure. Mm-hmm. And we use non-human primate model as to understand how we can cure this infection. Yeah. So we've been doing a lot of what we call immuno-based intervention, targeting the immune system to better recognize and target the virus. Mm-hmm. And at the beginning of the pandemic in 2020, we felt that we really need to use our experience in the monkey model to adapt the model for SARS-CoV-2 infections.
Right, yeah. So when this pandemic unfolded, of course, we knew very little about this virus. We had no vaccines. We had, other than some really generic drugs, over-the-counter drugs that can help with some symptoms, we didn't really have any good drugs or therapeutics. And so that is where your lab stepped in?
Yes, so a lot of people at the beginning were working on vaccine. Mm-hmm. As I said, our experience is more on immuno-based intervention. So if you remember the beginning of the pandemic, the situation was dramatic in Italy. Yeah. Mm-hmm. I remember I was watching on the news of a military truck. They were taking away body from city because the cemetery were too full. Yeah. And I was talking with some clinician in Italy, and they were saying, we don't know how these people are dying, but we do see very high level of inflammation and cytokines. Mm-hmm. So I was starting thinking to use a compound that can reduce inflammation. And I had the midnight call from Ray Shinazzi that was working with this compound called baricitinib, mm-hmm. that is approved for rheumatoid arthritis to reduce inflammation. And he's very interested to test also in the context of HIV infection. So we started discussing the idea to create a monkey model to test baricitinib, and this is what we did.
So when we talk about a severe inflammation in diseases such as caused by SARS-CoV-2, it is ultimately your own immune system that kills you.
Yeah, so when you have a pathogen, can be cancer, can be infection, virus, etc., your immune system, of course, fight this pathogen. Mm-hmm. And to do this, it creates inflammation that is beneficial because the immune system can clear the infection. Yeah. But in many conditions can be also detrimental, in particular if it's too high or if it's not controlled, it's prolonged. Mm-hmm. This, for example, happens in HIV infection because you don't clear the virus, so your immune system is always what is called chronically activated and exhausted. And in the context of SARS-CoV-2 infection, it's been clear from the beginning that people that were dying were having very severe inflammation. So your immune system is so activated, in particular your T cells, your macrophages, your neutrophil, that were producing a compound that were actually detrimental to the host.
And so to get back to the research you did, so you use a primate model, so these are monkeys, and so you infected these with SARS-CoV-2 and then you tried these compounds on them?
Correct.
And how did that go for the monkeys?
Yeah, so we infected a group of control animals just infected with mm-hmm. SARS-CoV-2 and a group of animals that were infected, and also we start treatment with baricitinib starting 2 days post-infection until 10 days. And we actually have two, I think, very clear black and white results. One, that there was no impact at all on viral load. So the amount of virus that was replicated was exactly the same, where there was a very potent impact on reducing inflammation and the pathogenesis. And these animals have not only much less inflammation, but as I said, less pathogenesis, and this was the main results of the study.
Yeah. So you can get rid of the symptoms by reducing inflammation, but it didn't work against the actual virus. So if I think about that, if you were to think about this in humans, would that mean that the patient would be fine but could still spread the virus to other individuals?
Correct. So this study actually very important clinical impact because baricitinib has been tested in people infected with SARS-CoV-2 and is actually present the only immunomodulator that is FDA approved for severe COVID. So other drugs are antiviral, targeting directly the virus replication. Baricitinib will target the immune system. So there is a lot of people that have been treated with this compound. Indeed, when we published the study, coming to your point, we proposed, because at that time there was no data yet in human, that potentially the best intervention may be a combination of an antiretroviral that will reduce viral load together with baricitinib will reduce inflammation.
And so this is happening in the clinic now, and so that goes back to your study to demonstrate this works in monkeys, and now we're using it for people.
Correct, and I think the advantage of baricitinib is that it was already approved for rheumatoid arthritis, again with the idea to reduce inflammation, so it was easier to translate immediately in clinic. And as you said at the beginning, inflammation is involved in many diseases, and not only diseases, including aging, etc. There is now inflammation is considered one of the key causes of many complications in human health. So actually baricitinib may apply to many other conditions. Indeed, clinician, Rechin Atsier-Temor and others are testing baricitinib in the context also of people living with HIV. With the same concept, there is very high level of inflammation. The path to this inflammation are very similar to the one described for COVID. So the idea is that baricitinib may be highly beneficial also in the context of people living with HIV.
Yeah, so it sounds like the more you learn, the better the therapeutics will be. And so maybe as a final question, how will that prepare us for the next pandemic?
Yeah, so I think we learn a lot, right, with SARS-CoV-2, and it's been, I think, very important to see how many groups come all together with, I think, a very highly collaborative spirit. And there is a lot we can do better, but I think it's been a really important lesson. And from our side, the Primate Center, I believe, has been very successful to, again, use all our experience to adapt to this new virus. And it's not only new virus, but was also a new condition. You need to use what is called the animal BSL-3 level to maintain and infect this animal. So there's been a lot of training for people at the Primate Center, etc. Yeah. There's been a lot of work, of course, in developing assay to quantify the virus. There has been a lot of, as you know, work to develop vaccine and therapeutics. So I think all of these should make us ready for the next pandemic. And of course, one thing very important will be to have, and this is happening, to have set apart funds to be ready from day one to start with the study without waiting for other funds or NIH, etc. And this is something that Temor is investing a lot on.
That's wonderful. That makes me feel better. I feel more prepared for the next pandemic now.
Hopefully, we are right.
Yeah, well, thank you so much for your work, and thanks for your time and coming on this show. And I want to thank you for watching. I hope you enjoyed it, and we'll see you next time.